Well, good morning, everyone. Thank you, Lars, and to the other organizers for giving me an opportunity to tell you a little bit about the work that we've been doing. And what I want to do is provide an example of how we're trying to combine data mining and text mining to build a reference knowledge base about cell phenotypes. So as Lars mentioned, I'm the new scientific director at the National Library of Medicine. NLM is one of 27 institutes at the U.S. National Institutes of Health, and we manage a large number of different data and knowledge resources for the research community, including things like GenBank and SRA, PubMed Central. And I think our goal is really to provide a trusted source of biomedical information for the research community. So as one example, this is the gene expression omnibus, GEO, that hosts gene expression data. So there's about 200,000 experimental data sets that are available in GEO for people to be able to query, download, and analyze. And so this is a really nice resource for the community if you're interested in understanding gene expression patterns in different kinds of experimental settings. And so this is an example of one of the data resources that we manage at NLM. We also have a really important knowledge resource, and that's in the form of PubMed and PubMed Central, where we have knowledge that's reported in the scientific literature. Now, the only problem with that is that the knowledge is embedded in free text. And so if you really want to use this knowledge for computation, it can be quite challenging, and that's where techniques like natural language processing and text mining come into play. So what we're trying to do at the National Library of Medicine is to come up with strategies that allow us to kind of bring together the data and the unstructured knowledge into a form of computable knowledge, in this case in the form of a semantic knowledge graph. That will allow us to kind of combine data mining and text mining to build a knowledge resource. And the example that I'm going to provide today is efforts that we're embarked on to build a cell phenotype knowledge base that we're tentatively calling NCBI cell. So the reason that we're interested in building a cell knowledge base is that there doesn't exist one at the present. And also that cells are really the most fundamental functional unit of life. We all know that different cells play different physiological roles in our body, and that the function and the phenotype of the cell is dictated by that subset of genes that are expressed in that individual cell. And any kind of abnormality in the expressed genome is really the physical basis of disease. So we're very interested in trying to understand cellular phenotypes, and historically, we've used transcriptional profiling of bulk samples to try to understand which genes are being expressed in which samples and which cells. But the problem with that bulk analysis is that it obscures the contribution of rare cell types that might be present in that sample. And so fortunately, over the last decade or so, we've seen a revolution in the ability to actually perform transcriptome analysis at the single cell level using single cell RNA sequencing. And so now this is allowing us to evaluate both known and novel cell types and identify rare cell types within complex tissues. So in parallel to that kind of laboratory revolution, we're also now seeing a revolution in the computational methods that we can apply to these data, including the application of artificial intelligence techniques. And I'm going to provide you an example of how we're using machine learning, but especially explainable artificial intelligence approaches, because the explainability helps us learn more about the biology that's behind these machine learning models that we're building. So I did a PubMed search to look for publications using single cell RNA sequencing. So there's close to 14,000 publications using this laboratory technology. And even within the last year, if we just look for full text articles that are freely available through PubMed Central, there's close to 2,000 publications just within the last year. So the amount of data that's coming out of this kind of technology is really expanding pretty dramatically. And so there are a lot of resources that are managing this kind of single cell data. The sequence read archive that I mentioned earlier is one place where you can get access to the primary read level data. But then there are also other repositories like the cell by gene single cell data repository that's been developed by through the Chan Zuckerberg Initiative that is managing some of the process data that derives from the analysis and processing of the sequence data. And so this is a really valuable resource if you're interested in starting out with an expression matrix as your starting point for analysis. So the workflow that we use to perform single cell RNA sequencing is fairly standardized now. You take a piece of tissue, you isolate either single cells or single nuclei, and then you do cDNA synthesis and amplification and sequencing of that material. And then you process the read level data to ultimately produce what we call a cell by gene expression matrix. And that's what you're seeing over here in this kind of heat map representation, where each column corresponds to an individual cell and each row corresponds to a gene with the color corresponding to the expression level for that gene in that cell. And then typically what we do is we want to group together cells that have similar transcriptional profiles, and we do that by using unsupervised clustering. And so that's what you see over here on the right-hand side, where we've taken this and we basically grouped all these cells together into a single cluster because they share a very similar expression profile. And so we view these transcriptional clusters that come out of this analysis as representative of either cell types or cell states. And I frequently use cell phenotype as a way of kind of combining the two. These are the genes that are expressed in these cells under these conditions. So this is an example of a study that we did looking at the middle temporal gyrus in the human brain. We microdissected that region of the brain out, and we did single nucleus RNA sequencing on that material. And we did the unsupervised clustering and came up with 75 different types of cells in that region of the brain. And so over here on the right-hand side, you can see a heat map here. And at the top is a dendrogram that kind of where each column here corresponds to one of these 75 cell types. And the dendrogram reflects the relationship between the different cell types that was derived using hierarchical clustering. And so you can see that there are three main branches here. There's a non-neuronal branch, and then there's a branch for, excitatory neurons here, and a branch for inhibitory neurons over here. In the heat map, we see kind of a reflection of known biology. These are marker genes that the neuroscience community has previously defined as being good markers for certain types of neurons and non-neuronal cells in the brain. And so in the first row there, you see a gene called GAD1, which is a marker of inhibitory neurons. And you see that all of those cell types kind of on the left-hand side of the dendrogram, they all express GAD1, indicating that they're inhibitory neurons. And then kind of in the middle of the dendrogram, you have cells that express another marker, SLC17A7, and that's a marker of excitatory neurons. And so based on this kind of known biology, we can start to categorize and characterize these different cell types. But what you'll see is that, so they're kind of in the middle, there's the VIP1 branch where all the inhibitory neurons express this VIP1 protein. But that's about all we know about the distinction between subtypes of VIP1 inhibitory neurons. We didn't have any idea about the subtypes and what marker genes they might express. And so what we wanted to be able to do is use machine learning approaches to try to define very specific marker genes for each of these different cell types. And so this was work that was done with my group, Brian Averman, Angela Liu, Beverly Peng, and Renee Zhang, to try to use random forest machine learning to discover minimum marker gene combinations that could be precisely define each of these different cell types. And so this is essentially the way that the algorithm works. So we start out with the cell by gene expression matrix that's been subdivided into these cluster groups by unsupervised clustering. And then we build random forest classification models to distinguish cells of one group versus cells from all the other groups. So it's a binary classifier that we're building. But really what we were interested in is not so much the classifier itself, that model, but the features that were most useful in making the distinctions between the different cell types. So out of the random forest output, we actually get a ranked list of informative features that help distinguish the different cell type clusters. We were interested in identifying features that have a binary expression pattern so that they're expressed at high levels in the target cells and not at all in the off-target cells, so kind of an on-off expression pattern. And so we re-ranked the informative features based on a binary score that reflects that kind of quantifies that kind of a pattern. And then we take those candidates and we build a signal decision tree using various combination of those markers. And then we select the optimal combination as the combination that gives us the best classification performance based on F-beta score evaluation. So that's the way the algorithm works. And this is what the results look like for those 75 different cell types. And so what you want to see is this nice, clean, diagonal expression pattern where you have individual gene or gene groups that are only expressed in cells of a given type. And so on the right-hand side, I have an example where I kind of blown up that region of the heat map. And I wanted to highlight an important feature of this approach. And that is that in contrast to just looking for genes that are differentially expressed, we're actually looking at the combinatorial pattern of the expression. And so in the box that I've highlighted in red, you see two genes that are expressed in that one VIP expressing inhibitory neuron subtype. You can see that the top gene is also expressed in a neighbor cell type on the left, and the bottom gene is expressed in a neighbor cell type on the right. But the only cells that express that combination of markers are cells of that one cluster in the middle. And so in this way, we use this combinatorial feature as a way of characterizing each of these different cell types. And then the other thing that I think is really interesting that is kind of emerging from this kind of analysis is because we're using explainable AI and we're keeping track of the features that are informative for classification, we're learning about the underlying biology, about what it means for cell type identity. And so if you look at the arrows that I've highlighted on the right here, these are all long non-coding RNAs. And so what we're finding is that many of the very specific markers that seem to be involved in cell type identity, at least for neurons in the brain, are long non-coding RNAs. And it was a big surprise when we made this finding. But I think it suggests that there's probably a second layer of regulation. We think a lot about transcription factors controlling the initiation of transcription, but there's probably an important second layer of regulation that involves long non-coding RNAs that seems to be very important for maintaining cell type identity. So I think this is just one example about how some interesting new biology can kind of emerge from this kind of analysis. The other thing to point out is that and we've applied this technique on a lot of different cell data sets from a lot of different organs. And it seems like on average, 2 marker genes is sufficient to give us maximum classification accuracy, regardless of whether we're dealing with neurons in the brain or cells in the lung or other tissues. So there are other techniques that people have used to pull out marker genes, and we actually can compare the performance of these other marker gene selection methods using the F-beta score for classification accuracy as our performance metric. And what we find is that NS4 kind of consistently produces better marker gene combinations than other methods on the same data set, the same cell types, based on this F-beta classification score. So you can kind of see on the x-axis here is F-beta from NS Forest, and on the y-axis is the F-beta from these other approaches. and we routinely get better performance using this approach. So we can use these marker genes for a number of different applications. Like if you're interested in doing spatial transcriptomic analysis to understand the spatial relationships between different cell types, you can use these as probe sets to be able to look at these kinds of spatial relationships. But what I want to do is show a specific example of how we can take this data and translate it into a semantic representation about cell phenotype knowledge that then we can incorporate into tools like graph databases and those kinds of representations for exploring the knowledge that is kind of derived from processing these data. So this is kind of an example where now we take the marker genes from the cell types in middle temporal gyrus, and now we can convert that into a semantic network graph. And in this case, what that allows us then to do is understand the relationship between the different cell types that reflect the hierarchical clustering result. So we have, in this case, on the top part of the graph are all of the GABAergic inhibitory neurons, and then the different subtypes with kind of satellites of sub-subtypes kind of revolving around the individual subtypes. And those are in the kind of the pink color. And then the marker genes are in the yellow color. So now we can take the data and convert it into this semantic representation about cell types, their relationships to other cell types, and the expression of marker genes that seem to be important for cell type identity. So once we have that, then we can start thinking about integrating that information from the single cell data into other sources of knowledge. And so in this case, on the right-hand side here, I have, in this part of the graph, this is the representation of the data that's coming out of the single cell experiments. And on the right-hand side, we have information about gene disease associations that might be coming out of genetic association studies. We have information about drug-drug target interactions that might be coming out of binding studies. And we have information about protein function that might be coming from the gene ontology and places like that. So now we can start thinking about integrating the single cell data with all this other source of knowledge that are being managed by other repositories of information, many of which are managed by the National Library of Medicine, but some are from other resources as well. And at the end of my talk, I'll show you an example of what can emerge when you actually do this kind of an integration of the knowledge. But this is a text mining session, and so we have this one source of knowledge that can come from data mining, but what about all this knowledge that's embedded in the literature? Can we extract that using text mining and integrate that into this kind of knowledge representation? And so that's kind of the second goal of the project, is to be able to take the text in the documents, translate that into what we call structured semantic assertions, where we're capturing subject-predicate-object relationships, and then integrate that into this knowledge resource. And so now we have two sources of knowledge, one coming from data mining and one coming from text mining. So that's the goal. So this work is being spearheaded by a postback in my group, Noam Rottenberg, in collaboration with Robert Lehman, who's working with Zhang Lu. We are learning a lot from this interaction with Zhang Lu's group and with Robert. And I think it's going to be a really interesting collaboration where we're bringing together people with expertise in data mining with people with expertise in literature mining. So I think the goal of this part then is to be able to go into the scientific literature, identify mentions of cell types using named entity recognition. But the challenge is that we're now seeing mention of known cell types as well as novel cell types that are being defined in these kinds of single cell experiments. So the first challenge is to be able to extract information about novel cell types from the literature. The second is, once we do that, we want to capture the relationships between cell types and other kinds of priority entities that are described in these publications, like marker genes, expressed gene sets, gene variation effects, enriched pathways, gene products, drug binding, disease associations, et cetera. So we're seeing a lot of reference to these kinds of important features in these papers, and we want to be able to extract those relationships as well. And then finally incorporate that into this cell graph knowledge-based prototype that we're developing. And we think that the text mining of the literature is going to be very complementary to the data mining pipeline. A lot of the single-cell RNA-seq data is not always freely available. A lot of it is available in some of these resources, but some isn't. So we could still get access to that knowledge by looking in the literature. We can also incorporate knowledge from other experimental platforms like flow cytometry, where we've learned a lot about cell phenotypes, and proteomics and metabolomics that are not using single-cell transcriptomic analysis. And then finally, the other part about the information that you find in the literature is that the authors, as subject matter experts, focus their discussion on the things that they think are most relevant from a biological, biomedical perspective. And so by doing text mining, we're able to actually kind of get the information that the subject matter experts view as being the most important. So we wanted to start out with the named entity recognition to be able to identify mentions of cell types in these papers. And so we basically took three kinds of approaches. We started out by using a supervised deep learning approach, where we started out with the Biomed-BERT foundational model, and then we fine-tuned that using several existing annotated training corpora, where people have gone into papers and they've pulled out these entities that would be interesting and relevant. And I think probably several people in this room have been involved in generating some of these corpora, which are extremely valuable to do this kind of work. The second approach was to use a tool called Phenotagger that Zhang-Lu's group developed to identify phenotypes in the literature. And then finally, we can take the results from these different classification models and merge them together using ensemble strategies to try to get the best of all these models in an ensemble result. So these are the four corpora that we used to train the Biomed-BERT model. I'm not going to go through all the details here, but to note that we selected these particular corpora because in every case they identified cell types as one of the entities that they annotated for these text resources. And they each have their different kinds of design details that I think are somewhat complementary. And so it would be worthwhile to see how they each can perform independently. And so then basically what we did was we took Biomed-BERT and fine-tuned it with these different corpora of annotated text to develop 4 separate models. And the reason that we started out with Biomed-BERT rather than other kind of flavors of BERT is that Biomed-BERT was specifically trained from scratch on biomedical text. And we think that that actually is especially important for the tokenization step. And in the example that I have here, you can see the impact that the different models have in how the data is how the text is tokenized. So if you were just to use the garden variety BERT and look at how cardiomyocyte was tokenized, it's divided up into 5 tokens because using general language, you don't really recognize this complete term as being something useful. Whereas if you look at how it was tokenized in Biomed-BERT, it's maintained as a single token. So we think that using these models that are specifically trained on the relevant text is a much better way to go than these more generic models. So then in terms of the cell Phenotagger, sorry. So this Phenotagger is an approach That's kind of a hybrid method that combines both dictionary and machine learning based methods to recognize ontology concepts in unstructured biomedical text. And the original version of Phenotagger was trained using the human phenotype ontology. And so it's designed specifically to be able to capture information about human phenotypes. But we're interested in cell phenotypes. And so what we did was we took Phenotagger and retrained it using ontologies that are relevant for our purposes. So we used the cell ontology and provisional cell ontology to identify cell types, and then Uberon to identify anatomy terms. And so on the right-hand side, basically what we do is we use the vocabulary source to provide positive examples for our training data. And then we go into publications from PubMed Central, and we pull out 40,000 n-grams from that text. And then we filtered those to remove any that had the ontology terms from our positive examples. And so that gave us now our negative examples for the training data. And so then we used that to train Biomed BERT, and then we came up with cell Phenotagger that's been tailored specifically to be able to recognize cell type related terms. So this is all really new data. It's only a month or two old, but already I think we're seeing some really promising results in applying this kind of strategy for extracting information about cell types from the literature. So here's the performance results of the 5 different models that came out of this fine-tuned training. And we looked at very strict exact matches of cell type phrases. And we also looked at approximate matches where we got partial matches of the cell type phrase. So, the examples that I give here is an exact match might be myeloid-derived suppressor cell is extracted, and that matches to myeloid-derived suppressor cell in the training corpora. And then the approximate example is where we have suppressor cells extracted, but really in that text, there was myeloid-derived suppressor cell was the phrase that refers to the cell type. So, I think we're seeing pretty good F1 performance on the approximate matches, lower performance on the exact matches. And so we want to kind of explore that in a little bit more detail to try to understand why are we missing some of these exact matches. But before I go to that, the other thing we wanted to do is combine the results from these 5 different models using an ensemble strategy to see how well the ensemble would perform. And so now here's a precision recall curve where we're looking at both the strict matches and the approximate matches using just basically taking the proportion of results that include the correct match from the 5 different models. So it's a pretty simple way of generating an ensemble result. But it's looking pretty interesting, pretty good. Even with the strict match criteria, we're getting an area under the curve of 0.82. So I think there's a lot of promise in the approach based on these kind of early results. So here are some examples of what the results look like. So some examples of true positives, we can recognize things like dermal fibroblasts, regulatory keratinocytes, tumor infiltrating lymphocytes. We can get strict matches of those things using this approach. There's some interesting false positives where we're kind of matching to anatomical structures instead of cell types. There are some false negatives where we're not able to detect some kind of unusual abbreviations for cell types, like MQ in this one paper was used as an abbreviation for macrophage. I'm an immunologist. I've never seen MQ used to abbreviate macrophage before, so maybe it's not so surprising that the method didn't pick that out. But I think the bigger issue is that this incorrect span, where we're getting part of the phrase, but not the complete phrase. And I think this is going to be really critical to be able to solve this problem, because when groups are defining novel cell types using single-cell RNA sequence data, they're typically describing them using this kind of a syntax, where the second part of the phrase is a known cell type, and then the adjectives that come before that core term are the new features that are defined from these experiments. And so I think one of our goals in moving this project forward is to be able to extend the recognition of these cell type phrases to include these upstream adjectives that are being added into the nomenclature coming out of these kinds of experiments. So in conclusion, we're seeing, reasonably good performance of the named entity recognition task, especially for core terms and classic cell types. I think we're doing a really good job of recognizing those things. And that if we pool the models together, we get, additional improvement in performance. In terms of the cell phenotyper, we're catching most of the classical cell types, but not the novel ones. And I think this is going to be one of our challenges about how we can address that challenge. And I mentioned that the most common error seems to be that we miss subtype descriptive adjectives that are being added to these cell type names. Some of these descriptors are relevant, like activated cells, and others may not be relevant, like classical cell. I mean, that's really the same as the cell type. So we're also going to have to try to make those distinctions. So the results that I just showed you, the test data was just derived from 10 publications that we manually annotated. So we need to expand that to a larger group to do a much better kind of testing of the results. But we did select publications that are recent that included the description of novel cell types. And those are things that were not present in the training corpora. So we felt like this was a better way to test it because these are the kinds of things that we want to be able to extract. In addition to applying the method to the free text, we also want to be able to apply it to the supplementary materials of papers, like the tables and even maybe the figures that have a lot of knowledge embedded in them that I think could be quite valuable if we can figure out how to extract it. And then we have to start working on relationship extraction, which I think will also be a pretty significant challenge. But so ultimately what we envision is that we want, is that me? We want to build this knowledge graph, a semantic knowledge graph that's kind of in the center of this diagram here. And there are two sources of knowledge that we want to try to bring in. One is there are these repositories that have all this data coming from, like in this case, the single cell transcriptomic community. We want to process that data through standardized data mining pipelines. Data mining pipelines to be able to produce interesting phenotypes about the cells that are being identified in these experiments, like biomarkers, like differentially expressed genes, like enriched pathways and biological processes. And then we can translate that into these semantically structured assertions to be able to load into that knowledge graph. The other source will be from text mining to be able to use different kinds of language models and things like that to be able to extract similar kinds of semantic assertions from that source of knowledge. And then when we do that, we can then think about integrating all these other knowledge resources into this to try to understand the relationships between cells and genes and diseases and drugs and things like that. And then to build a user interface to make this information accessible to the community. This is going to be another one of our challenges because the vast majority of our research community is not going to be comfortable processing information in a graph database, right? They're going to need some kind of user interface that allows them to access this information. And so that's going to be one of our challenges. But I have one example of the kinds of things that can emerge once you develop something like this. So here is an example where we have taken our cell types from the middle temporal gyrus. We've integrated that with information about drugs, genes, diseases, genetic predispositions. And then in this case, what we did was we said, okay, I want to pull out a subgraph from that network of edges that link the compounds, diseases, genes, and cell types with Alzheimer's disease. So we have Alzheimer's disease in the middle here. And now here are all the connections that come out of that extraction of that subgraph. But as an example of things that I think are potentially interesting that come from this kind of analysis is that this is a particular type of inhibitory neuron that was in the middle temporal gyrus. It has one marker gene that specifically defines that cell type, this gene chRNA7. And it turns out that chRNA7 has both been found to have a genetic association with Alzheimer's disease in GWAS studies. And it also is, the protein is bound by a drug, galantamine, that is being used in trials to treat Alzheimer's disease. And so I think this is the kind of information that can come out of these knowledge graphs that now start us to learn about things like maybe potential drug targets and other kinds of associations. So let me just end by, we have a series of publications about these methods. All of our source code is available in GitHub. We try to incorporate a lot of this knowledge into the cell ontology and the provisional cell ontology for people to be able to use. So those resources are available. And then finally, I acknowledge the people who were involved in most of the work, but we've benefited from collaboration with a large number of different groups, especially Ed Lean's group at the Allen Institute for Brain Science, where we've collaborated on all of the brain analysis work. And then our funding sources over the years, including the National Institutes of Health that's funding the current work. So with that, I will stop, and I'm happy to take any questions.
All right, we have time for one or two quick questions, and please use the microphones.
Thank you. Really exciting work. I have a very quick one and then a more complicated one. The very quick one, with the improvements, with Robert's involvements and the improvements in cell annotation, do you expect that these annotations are going to find their way into PubTater, which I think has cell lines, but maybe this would expand that?
I hope so.
Okay.
Yes.
Awesome. And then the other one is for the knowledge graph-based approaches. It seems that the method of cell marker selection is relatively strict, even if those gives you very good results.
Yep.
And so I wonder if there's, if you could comment on whether that is too strict for when you start linking it back to function, because you may have things that are inferior markers, but still very functionally important.
Yeah. No, I think that's a great point. And we decided to start out with this very strict connection between markers and cells, but there are also differential expression analysis where you might have 20, 30, 40 genes that also have reasonable differential expression that probably play an important functional role in the cell. So we are starting to think about how do we kind of bring both types of information into the knowledge graph. How do you quantify the edges? So you bring in some evidence, some level of confidence and evidence for the assertions that are being made in those connections. So there's a lot of work along those lines that still needs to be done. But yeah, thanks for your question.
Okay, one last question over here.
Thanks for the nice overview, Richard. I have a question on the combination of the single-cell RNA sequencing data and the phenotyping.
Yeah.
Because now you're just looking like in a cell ontology. But if you think about more general phenotyping of a cell, I could have a dendritic cell that is in an inflammatory state and it is migrating, right? So you could get a lot of that information from the transcriptomics data. How do you anticipate combining the transcriptomics data for then better cell states and type
I think that's one of the reasons why we're excited about going into this text mining from the literature, because a lot of those kinds of phenotypes are described in the publications but are not apparent in the single cell RNA sequencing data. So this would be a way for us to link those things together. The challenge is that these transcriptional profiles provide very precise definitions of cell types, and in a lot of the experiments, people are not doing the experiments at that level of granularity. So we're not always able to connect the phenotype from the experiment with these very granular cell types that are in the single cell data. But we can connect them to a parent cell type in the ontology. So these are the kinds of things we're thinking about.
Thanks.